On the cover, you can see some images of brains, some neuroimaging pictures here. We'll go over that near the end of the chapter and how that relates to psychology. You may be familiar with a lot of the science that we're learning today, but your book does a really good job of connecting these biological processes to behaviors and psychological disorders. So definitely go ahead and read the textbook before you watch this video. And when you're done, come on back and watch the video because I have lots to explain. Let's get started with human genetics. Psychological researchers study genetics in order to better understand the biological factors that contribute to certain behaviors. So that's why we're talking about this today, because in psychology, we want to understand the biological factors that cause or contribute to behaviors or behavior disorders. Some of the basics that you need to know are chromosomes are large strings of genetic material. This genetic material is known as DNA. In each chromosome, sequences of DNA make up genes that control or partially control a number of visible characteristics known as traits. So your genes control your traits. The traits are what we see. An allele is a specific version of a gene. So for example, a gene that codes for the trait of hair color can have different alleles affect which hair color the individual actually has. So the allele is the specific version of the gene, like the brown hair version of the hair color gene. A genotype is your genetic makeup, whereas your phenotype are things that we can see. They're the inherited physical characteristics, which are a combination of genetic and environmental influences. And we'll talk about that combination in a moment. But first, let's look at the diagram or the picture on the right to understand better that a genotype is always your DNA. In the top example, your genotype is DNA and your phenotype is your thumbprint or your fingerprints. Your body organizes physically based on what the genes tell it to, based on how the genes tell it to organize itself and result in a phenotype of a thumbprint. The orange example has your genotype still being DNA, but this time the phenotype is height. So your body will grow or fail to grow based on what the genes say, how your genes tell it to, what your genes tell it to do. So again, genotype is your genes, your DNA, and phenotype are the physical traits that we can see. I like Punnett squares a lot. I think the book does a good job of explaining it, but I want to go over it real quick. It shows a real simple explanation of genetics and how it works, where the inside squares are the possible offspring, and along the top we have the fathers or the male plants, and down the side we have the mothers or the female plants. Of course, this works for people and animals as well, but it's often simple to look at an example in plants. Let's look at the example on the left, where we have a green bean and a yellow bean mating, right? Basically having or creating offspring, right? And being yellow is a recessive trait, meaning that you need to have both of those lowercase yellow g's in order to actually appear yellow. So while we can see that the mother does carry that recessive gene, that lowercase g, because she has the uppercase G, which is the dominant trait, she appears green. The male plant, however, appears yellow because he has both of the recessive, a homozygous allele with two recessive lowercase g's. So for that reason, the male plant appears yellow and the female plant appears green. Now, each of these parent plants will donate one of their alleles to their offspring. So within the square, you can see the four possible combinations that their offspring can have. In this case, it would be a 50-50 chance if their offspring end up green or yellow, since there are two options that result in green appearance and two that result in yellow. But if we look at the flower example over on the right side, we can see that both parents appear purple, and that's because they both have an uppercase and a lowercase b. They both carry that recessive gene for whiteness, for the flower being white, But because they have their dominant uppercase B purpleness gene, they both appear purple. When we look at the combination of the male and female plant possible offspring, we can see there's a 1 in 4 or 25% chance that their offspring will appear white, and a 75% chance that their offspring will appear purple. So this is why in the human world we see that you might have a child with blue eyes whose parents both have brown eyes, and the reason for this is that the two brown-eyed parents are carrying the recessive blue-eyed gene. And if you end up in that 25%, you'll end up with blue eyes. However, eye color has been found to actually be polygenic, controlled by more than one gene. So it's a little more complicated when we look at the Punnett square. I'm not going to bother going through all of the statistics of this. I thought about maybe having an assignment on Punnett squares, but it's a little too math heavy for a psychology class. Decided to let it go. If you're interested in learning more about Punnett squares, though, please contact me because I do have a lot of fun resources and worksheets about Punnett squares. Moving on to the interaction between genes and our environment. I think it's kind of common sense that who you are is an interaction of your genes and your environment. Some people refer to this as the nature-nurture issue. However, different researchers and different scientists have different perspectives on how this interaction works. We'll talk about 3 of these main perspectives. The first is a very interesting one called range of reaction. Range of reaction tells us that our genes set the boundaries within which we can operate. So they set the range, right? The highest end and the lowest end. And then our environment interacts with those genes to determine where in the range we will fall. I really like this example, so let's take a look. In the top red row, we can see that these people were raised in an enriched environment. Perhaps they went to good schools, their parents read to them, they had a good diet, nutritious foods, they didn't have to worry about their survival, all of that enriching environment stuff. Excuse me. So while each person is born with their own possible intelligence range, you can see that Tom, Kimberly, and Ted all ended up falling near the top end of their range due to their enriching environment. Moving down to the yellow average environment people, their red dots ended up somewhere in the middle of their range because they were raised in an average environment in terms of realizing their intellectual potential. Down in the deprived row, we see Jack, Jill, and Alice, who have all ended up near the bottom of their ranges. So those are the intelligence ranges they were born in. Let's say we took Alice and moved her up to that enriched environment. She may have ended up with the highest intelligence of the whole bunch, because look at that big range she has. She could be all the way up in genius level intelligence. But because Alice was raised in a deprived environment, she fell near the lower end of her range and ended up a couple points below Ted, who does not have nearly the same intelligence potential as Alice does. But because of his upbringing, he ended up scoring a little bit higher. For another example, let's compare Tom at the top and Jack at the bottom. Their ranges are almost, if not completely identical. They were born with the same genes, the same intellectual capabilities. But because Tom was raised in an enriched environment, he ends up with an IQ of 84, and Jack, raised in the deprived environment, ends up with an IQ of 66. And the difference between 66 and 84 may just be the difference in being able to hold down a job or take care of yourself as an adult. So as you can see, environment is very important, as are the genes that we're born with, right? Even though Tom was raised in that enriched environment, he still only has an IQ of 84 because that's as high as it's going to get for him. So that's range of reaction. When you're born with a particular range due to your genes, and your environment will determine where you fall on that range. Epigenetics, however, is the study of how the same genotype leads to completely different phenotypes. The epigenetic perspective is very different from range of reaction because epigeneticists believe that the genotype is not fixed or limited at all. There's no top range that you could get. that if you have the right influences and the right environment, it's almost like your genes don't matter. Not that they don't matter, but that you can really push it. There's no limit to how far you can go with the effective environment. Finally, on the right-hand side, my LeBron James example is called genetic environmental correlation. This tells us that our genes influence our environment, and our environment influences the expression of our genes. So we know that second part, right? The range of reaction tells us that our environment influences the expression of our genes. But the genetic environmental correlation theory also points out that the environment we're raised in is a result of our parents' genes, which we get half of, right? So in the LeBron James example, his son is a very good basketball player. He's a very talented, accomplished basketball player. And a lot of this comes from the genetics that he got from his dad, making him tall and strong and good at basketball. But the other part comes from his environment, being raised in a family that likes basketball, probably being put in basketball lessons or clubs at a very young age. Being exposed to a lot of basketball helped increase this young man's potential. It helped increase his talent because he was always surrounded by it, given opportunities to thrive in basketball. But the real important part of this is that the reason he was in that environment was his dad's genetics. So LeBron James's genetics, his aptitude for basketball, led him to create a basketball-rich environment for his family. It also led to him passing his genes down to his son. So LeBron James's son received those good basketball genes, and he received the basketball-related environment, both of which were due to his father's genetics. I hope that makes sense. The book gives a similar example about basketball, so you can read that as well. I do find this theory very interesting. Let's move on to some real sciencey science. We will talk about neurons, which are defined as interconnected information processors that are essential for all of the tasks of the nervous system. So the nervous system is made up of billions of neurons. The neurons are supported by glial cells. Glial cells do all sorts of things to help the neurons function. They provide the scaffolding on which the whole nervous system is built, right? The basis or the foundation to be built on. They also help neurons line up closely with each other. They provide insulation by making those myelin sheaths that you can see in the picture, the light blue covering. They transport nutrients and waste products and mediate immune responses. So glial cells are very important. They support the neurons, and the neurons are the information processors. They're sending messages, electric messages to each other all day and all night so that our bodies can function. Let's learn a little bit more about how. The neuron is the small information processor, and we can look at the picture of the neuron in the top right. It has dendrites on the end. You can see the dendrites at the very top. The dendrites are the input sites. That's where the signals come from the other neuron. So you can see very light up in the corner, there's another neuron with its myelin sheath. And it's that last neuron's terminal buttons are coming nice and close to the neuron that we're looking at and sending messages to its dendrites. So the dendrites receive the message, and then the signal, once it's received by the dendrites, is transmitted across the soma and then down the axon. The axon is the largest part of the neuron. It's transmitted all the way down the axon, ending at the terminal buttons. Those terminal buttons then send the message to the dendrites of the next neuron, and on and on and on until it gets to its destination, until the signal is done. So the terminal buttons contain synaptic vesicles that house neurotransmitters. The neurotransmitters are chemical messengers, and when the terminal buttons release those chemical messengers, they release those neurotransmitters into the synaptic cleft, which is the space between those terminal buttons and the dendrites of the next neuron. The neurotransmitters, let's look in this bottom picture here. So on the top part of this picture, we can see the terminal button of the last neuron. It has neurotransmitters that it's releasing into the synaptic cleft. And then the dendrites of the following neuron have receptors ready to attach to those neurotransmitters and continue the signal. So there's a little space between each neuron, but the neurotransmitters that are kind of thrown out into that space lock on to the next one and continue sending the signal. It's actually really interesting, I think, and really cool that this is all going on so many times a minute inside our own bodies. Let's look into this a little bit deeper and see just how it happens, how those neurotransmitters get into the next neuron. So when the neuron receives the signals at the dendrites that we were just talking about, those neurotransmitters from an adjacent neuron bind to its receptors. When this happens, small pores, or gates, if you will, open around the neuronal membrane. So around the membrane of the neuron, which is semi-permeable, meaning only sometimes certain things go through, the gates open up and those sodium ions go rushing in because they're pushed or propelled both by charge and by concentration differences. Your book explains this a little bit more in detail. So the sodium ions move into the cell, which makes the neuron become positively charged. Now, if that neuron becomes positive enough, if it gets high enough and crosses the threshold of excitation, the neuron becomes active and the action potential begins. The action potential is that electric message that we were talking about, that it's being sent. So it needs to hit a certain threshold. You need to get enough of those sodium ions into the neuron in order to trigger the action potential. In the top picture, we can see what happens during an action potential. And I kept the picture of the neuron there so we can continue to look at how things are traveling through the neuron. So if we look right at the beginning, let me zoom in for a minute. The last neuron was sending messages out of its terminal buttons using neurotransmitters. Those neurotransmitters attached to the receptors on the dendrites, causing the semi-permeable outer layer of the neuron to allow the sodium ions in. When the sodium ions came into the neuron, the overall charge went up, and once it hit a certain point, let's look at our action potential. Once it hit that negative 55, that threshold of excitation, the action potential began. And it goes up and up and up, and everything over that dotted purple line is considered the electrical message that's being sent. When it peaks, right when the positivity or the charge peaks, then all of a sudden the sodium gates close and the potassium gates open. So now we're letting the negative ones in. First we were letting the positive guys in, the sodiums. Now we're letting the negative guys in to bring it back down. This is called repolarization, right? It's bringing it back down to its resting potential of being negative. At first, it does something called hyperpolarization, which you can see brings it even more negative than it's supposed to be, just for a moment, and it goes back up to that resting potential, that regular negative charge that's sitting around waiting for the neurotransmitters to trigger the positive influx of sodium ions, triggering the action potential, sending the electric message along to the next neuron. And this happens over and over and over with an action potential happening in each neuron. So the action potential is the electrical signal that typically moves from the cell body, where the soma is, down the axon to the axon terminals, or the terminal buttons. The electrical signal moves down the axon with the impulses jumping in a leapfrog fashion between the nodes of Ranvier. The nodes of Ranvier are the little spaces in between the myelin sheath. If you notice, the myelin sheath, which was built by the glial cells, protects the axon of the neuron, but there are little spaces in between. At each point, some of the sodium ions that had entered the cell will diffuse to the next section of the axon, raising the charge past the threshold of excitation again and triggering a new influx of sodium ions. And this just keeps happening all the way down the axon until the terminal buttons, and then it passes on to the next neuron. I know this is pretty confusing. Don't worry if you don't totally get all of the biology of it, as long as you basically understand the order that things go in and what's going on with the charges. The book does a great job of explaining it, or feel free to reach out to me if you need more details on neuronal communication. The very last step in this is when it's done, when the action potential gets to the end of the neuron, to the terminal button. The synaptic vesicles release their neurotransmitters into the synaptic cleft, where they travel across the synapse or this empty space to bind to the receptors on the dendrites of the next neuron. Then the process repeats itself. One more important thing that happens here is called reuptake. So you can see that the neurotransmitters are being sent out into that open space, right? And some of them are being locked into the receptors of the next neuron, but there's some that are left over, just hanging around in that synaptic cleft. So reuptake is the process of the neurotransmitters being pumped back into the original neuron that released them. It clears out the synapse and sends them back to restart their job. All right, that's it for neuronal communication, but let's talk about the types of drugs that can affect neurotransmitters, because sometimes physical or psychological disorders can arise by having too many or too few neurotransmitters. So there are drugs that can act as agonists or antagonists for a specific neurotransmitter system. Agonists are chemicals that mimic the neurotransmitter. So they go sit in the receptor site, pretending to be the neurotransmitter, and they activate that action potential. It's sort of like a fake imposter neurotransmitter. Remember, the neurotransmitters are what trigger that action potential. They get the message started. And if you're not making enough of those neurotransmitters, you can take a drug that mimics the neurotransmitter and sits right in the receptor site to trigger that same process. On the other hand, an antagonist blocks the normal activity of the neurotransmitter. So he sits right there, the little red guy, in the receptor site, but this type of drug, it doesn't cause the reaction. It doesn't trigger the action potential. It just blocks other neurotransmitters from getting in there to trigger it. So this is when you have too much of a certain neurotransmitter. This is how you block it. You don't actually get rid of those extra neurotransmitters in your body. You just block the receptor sites so that they can't cause the action potential. So agonists and antagonists are both drugs prescribed to correct a specific neurotransmitter imbalance, right? They give agonists when there's not enough of a neurotransmitter and antagonists when there's too much. One other type of drug is called a reuptake inhibitor. And so if you remember, the reuptake is at the end of one neuron's job. There's leftover neurotransmitters in that empty space that need to go be sucked back into the neuron they came from. Reuptake inhibitors stop that from happening or make it happen slower so that your neurotransmitters have a longer time to do their job. An example of this is common antidepressants are serotonin reuptake inhibitors. So serotonin, which is a neurotransmitter that makes you feel good, they let the reuptake inhibitors allow the serotonin to hang out a little longer in your synaptic cleft, hoping that they will bond to the next neuron, right? So it gives them a little bit more time. It stops them from being put back into the neuron they came from too quickly. So that's it for the drugs. Oh, let's look at the different types of neurotransmitters real quick. You don't need to memorize these or anything, just some that you may have even heard of before and what our major neurotransmitters do in our bodies. There's acetylcholine, which can affect muscle action and memory. Beta endorphin, pain and pleasure. Dopamine affects mood, sleep, and learning. GABA is involved in brain function and sleep. Glutamate is involved in memory and learning. Norepinephrine affects our heart, intestines, and alertness. And serotonin can affect mood and sleep. So again, you can have too much or too little of these things, and certain drugs can help correct that. All right, now that we know how the neurons work within the nervous system, let's look at the whole system itself, right? We talked about how the actual messages are sent from one neuron to the other, but let's look at how this works as a system. The central nervous system is your brain and spinal cord, and we'll talk about that in a minute. But your peripheral nervous system is what connects the central nervous system to the rest of your body. The peripheral nervous system is made up of thick bundles of axons called nerves, which carry messages back and forth between the central nervous system and the rest of the body. Now, the peripheral nervous system can be broken down into two parts, the somatic and the autonomic nervous system. The somatic nervous system is associated with conscious or voluntary activities, things that you're doing on purpose or you know that you're doing, such as raising your hand or watching a movie. So it's involved in the relay of sensory and motor information to and from the central nervous system, right? Sensory information is sent from your body to your brain, and motor information is sent from your brain to your body. When you want to raise your hand, your brain sends the message to your arm to lift it up. This is sent through the somatic nervous system. The autonomic nervous system controls internal organs and glands and is outside the realm of voluntary control. These are just automatic, right? That's autonomic system. It just happens on its own. You cannot control it. Within the autonomic nervous system, we can split it up once more into the sympathetic and parasympathetic nervous system. The sympathetic part is involved in preparing your body for stress-related activities or something out of the norm. If I need to lift a box, my sympathetic nervous system will have all of the automatic processes in my body that account for that. Then the parasympathetic nervous system brings it all back to resting, right? It returns the body to the routine regular operations when it's not doing a stress-related activity. So as you can see, we have the nervous system in general, which is split up into two parts, the central nervous system, which has your spinal cord and brain, and the peripheral nervous system, which acts as the communicator between your body parts and your central nervous system. Within the peripheral nervous system, we have the somatic nervous system, which has our conscious or voluntary movements and our senses. And then the autonomic nervous system, which is the automatic, involuntary processes of our body, and that can be broken down into sympathetic and parasympathetic nervous systems. Now it's time to talk about the brain. So that was the peripheral nervous system. The brain and spinal cord are the central nervous system. I don't have anything in here about the spinal cord, but your book does a good job of explaining the basics. In this class, it's more important to focus on the brain, so that's what we're going to do. In the picture on the left, you can see that the cerebral cortex of your brain, which is the outer surface of the brain, has a lot of bumps, which are called gyri, and grooves, which are called sulci. And the sulci and gyri create more surface area on your brain because it's all folded like that, if that makes sense. And the biggest sulci of all, the biggest groove, goes right down the middle of your brain, and that one's called the longitudinal fissure. The longitudinal fissure is what divides up the two hemispheres of your brain. And in fact, the two hemispheres are connected by a band called the corpus callosum, which goes through the longitudinal fissure. And the book has a lot of good information about the two hemispheres of the brain and some research that has been done on it. For today, we're going to talk more about the functions of the cerebral cortex as a whole. Before we do that, let's look quickly at the thalamus, which is the sensory relay for the brain. All of our senses, except smell, are routed through the thalamus before being directed to other areas of the brain for processing. So it takes all the other sensory information, not smell, and routes it to the right part of the brain. The limbic system, which processes emotion and memory, also processes smell, which I find very interesting, that The thalamus is responsible for our other senses, but the limbic system, which is responsible for emotion and memory, is what processes smell. Personally, I associate smells with memories and emotions a lot. Within the limbic system, we have three main parts. The hippocampus, which is mostly responsible for learning and memory. The amygdala, which is involved in our experience of emotion and then tying emotional meaning to our memories. The amygdala is very interesting. And the hypothalamus, which regulates a number of homeostatic processes. So our regulation of body temperature, appetite, blood pressure, keeping everything level is the job of the hypothalamus. So these are some important parts of the brain, but as you can see in the bottom left, it tells us that the cerebral cortex, the outer surface of the brain, is associated with the higher level processes, such as consciousness, thought, emotion, reasoning, language, and memory. So let's look at that. And actually all of those things were in the cerebral cortex, and then soon we'll talk about the midbrain and the hindbrain. The cerebral cortex is divided into 4 lobes, 4 areas. The frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. And I have them color-coded over here on the side. This part is very important. It's probably the most important part of our brain lesson for today in terms of psychology. So let's look at what our different lobes do. The frontal lobe in orange, near the front of the brain, is involved in reasoning, motor control, emotion, and language. It contains the motor cortex, which plans and coordinates movement, the prefrontal cortex, which is involved in higher level cognitive functioning, excuse me, and Broca's area, which is involved in language production. In the book, they do a great job of explaining different circumstances of people with damage to their Broca's area and the symptoms that arise. Take a look at that part of your book for sure. Onto the parietal lobe in blue. This part is involved in processing the information from the body's senses. It contains the somatosensory cortex, which is essential for processing sensory information from across the body. This part's kind of interesting too because it's the way that it's mapped out in the parietal lobe sort of matches where your body parts are located on your actual body. The book tells more about that as well. It's quite interesting. The temporal lobe, which is associated near your temples, right, which are right above your ears on the side of your head. The temporal lobe is associated with hearing, memory, emotion, and some aspects of language. The auditory cortex is part of the temporal lobe, and it processes the auditory information, hearing. And then the Wernicke's area is for speech comprehension. And what's interesting is that in the frontal lobe, in that orange part, we have Broca's area, which is responsible for language production, speaking. Whereas Wernicke's area, over in the temporal lobe, is for speech comprehension. So depending on where brain damage occurs, some people may only be disabled in their ability to speak, or they may only have an issue with their comprehension, but not necessarily both, since producing speech and understanding speech are things that are two things done in 2 different parts of the brain entirely. I find that kind of cool. Finally, the occipital lobe is in the back, right above the brainstem. It contains the primary visual cortex, which is responsible for interpreting incoming visual information. So lots of good stuff here. Again, your book does a great job of talking about case studies of accident victims who have certain types of brain damage. And well, it's too bad for these people that they had to endure this pain and suffering and these behavioral circumstances. As researchers, it's really neat that we get to study a brain that's only been damaged in a certain area and see how that damage affects the person's behavior. All right, moving on to the midbrain and hindbrain. So remember let me go back real quick. The cerebral cortex is the whole top layer of your brain, and then beneath that, we have the midbrain, and then down at the back near your brain, near your spinal cord or your brainstem, is the hindbrain. So the midbrain, some important parts of the midbrain that we need to know about are the reticular formation. It extends up into the forebrain and down into the hindbrain. It connects them all, and it's important for sleep-wake cycles, arousal, alertness, and motor activity. We'll be learning a lot about sleep in the next chapter. The reticular formation is important for sleep. Also, the substantia nigra and the VTA, they produce dopamine, which is a neurotransmitter that helps with mood, sleep, and learning. These two areas are also critical for movement. And as you can see in a Parkinson's patient, the substantia nigra is very diminished, right? You can barely see that dark matter that used to be there, and that results in Parkinson's symptoms. Again, the book has more details about the Parkinson's stuff. Very interesting as well. Moving on to the hindbrain. The hindbrain is located at the back of the head. It looks almost like an extension of the spinal cord. Three important parts of the hindbrain are the medulla, which controls the automatic processes such as breathing, blood pressure, and heart rate. The pons, which literally means bridge, serves to connect the hindbrain to the rest of the brain. It's also involved in regulating brain activity during sleep. And the cerebellum, which controls balance, coordination, movement, and motor skills, and is important for processing procedural memory, the physical things, remembering how to do things physically. All right, now that we've learned about all the different parts of the brain and the nervous system, we can talk about how we know all of this, right? I discussed a little bit that brain damage patients are a huge window into the brain. It really allows us to learn a lot about the brain through brain damage patients. But another way that we can learn about the brain in bigger numbers and bigger samples is through brain imaging or neuroimaging. There are several types of neuroimaging. We'll go over the 5 main types. We have CT scans, which are basically x-rays. They take a bunch of x-rays of the person's brain or body and put those together into a composite picture that shows us basically an x-ray of the brain, or sort of more like a 3D x-ray. Then the PET scans, or PET scans, stands for positron emission tomography. These actually create pictures of the living active brain, not just the structure of the brain, but the active brain. So to do a PET scan, you need to drink or be injected with a mildly radioactive substance called a tracer. And once that goes into your bloodstream, the amount of tracer in any given region of the brain can be monitored. So basically they're telling how much blood flow there is in different parts of your brain, and the way that they can actually see this is through the tracer, the radioactive tracer that they pump into your bloodstream. As the brain becomes more active, more blood flows into that area, right? So we're monitoring the blood flow to your brain to see which parts of your brain have the most activity during certain behaviors or events. EEGs just provide the brain's electrical activity in general. It does not tell us the different areas that are activated or where the activity is happening within the brain. It just shows the general amount of electrical activity in the brain, but depending on the study, that can be enough. That can be all that you need. Over on the right side, we see that MRIs, magnetic resonance imaging, are when a person is placed in a machine that creates a strong magnetic field, and that magnetic field actually causes the hydrogen atoms in your body's cells to move. When the magnetic field is turned off, the hydrogen atoms emit electromagnetic signals as they return to their original positions. Doing this allows them to see the structure of your brain. And then functional magnetic resonance imaging, or fMRIs, use the same idea, but it shows changes in activity over time by also tracking the blood flow and oxygen levels. You don't need to know all of the nitty-gritty details of how these types of neuroimaging work. Just know that for the most part, fMRIs give us the most information in the safest way. And EEGs just give us the general amount of electrical activity, which can be enough depending on your research question. One more thing I'd like to talk about real quick is the endocrine system. The endocrine system consists of a series of glands that produce chemical substances known as hormones. I'm sure you've all heard of hormones, and I'm sure you all experienced hormones in your early teens. And in fact, we all continue to experience hormones every day and night. Hormones are involved in regulating all sorts of bodily functions. They're controlled through interactions between the hypothalamus in your brain and the pituitary gland in your endocrine system. And that's how they communicate with each other, is between the hypothalamus and the pituitary gland. And the hormones can affect lots of different parts of your life. I will show you a quick table of some of the important or major hormones that you have in your body and the glands that secrete them. So some that you might be familiar with include melatonin, epinephrine and norepinephrine, insulin, estrogen and progesterone, testosterone. These are all hormones you may have heard of before, and feel free to pause the video and take a look at this table and where they all come from and what their functions are in your body. Right? We probably all know that the pancreas makes insulin, which helps regulate blood sugar levels. And if your pancreas isn't producing the right amount of insulin, then you may be diagnosed as diabetic. We are ready for our summary. I know this chapter has a lot of sciencey science, but I hope you still found it interesting. Feel free to turn off the video and come back and watch this part right before you take your quiz. It's a good review. Let's do it. Chapter 3 summary. A chromosome is a large strand of genetic information called DNA. So large strands of DNA are called chromosomes. Your parents each give you chromosomes which contribute to your genetics. The DNA are actually helix-shaped molecules made of nucleotide base pairs. A gene is a sequence of DNA that controls or partially controls a physical characteristic or a trait. An allele is the specific version of that gene. The genotype is an individual's genetic makeup, whereas their phenotype are their physical characteristics that we can see. So your genotype is your DNA, and your phenotype is something like brown hair or blue eyes or being tall or having wide hips or big feet or anything like that that you may have inherited from your parents that we can see. Those are your phenotype. Polygenic means that a certain trait is affected by multiple genes instead of just one. That happens probably most of the time. 3 different ways of looking at the genetic environmental relationship. We have range of reaction, which tells us that our genes set the boundaries within which we can operate, and our environment interacts with the genes to determine where in that range we will fall. Remember the example with the intelligence ranges and their environments. Epigenetics is the study of gene-environment interactions and helps explain how the same genotype can lead to different phenotypes. Genetic environmental correlation, that's the LeBron James example. Our genes affect our environment, and then our environment influences the expression of our genes. But that environment is not random. It's affected by our parents' and grandparents' genes. Into this more scientific side of things of our nervous system, we have neurons, which are cells in the nervous system that act as interconnected information processors, which are essential for all the tasks of the nervous system. Neurons are the main players, but they wouldn't be who they are today if it wasn't for the glial cells. Glial cells are the nervous system cells that provide physical and metabolic support to the neurons, including insulation, communication, and nutrient and waste transport. Don't forget about glial cells. They're very important. Neurotransmitters are the chemical messengers of the nervous system. They're released from one neuron into the synaptic cleft, and then they get to the next neuron. They attach to the receptors of the next neuron. An action potential is the electrical activity that occurs at the threshold of excitation. It's an electrical signal that moves down the neuron's axon to its terminal buttons, where it causes more neurotransmitters to be released into the next synaptic cleft, and then they're picked up by the next neuron and they continue to send the message down the road. When the action potential, so I just said it, but let's read what it says here. When the action potential arrives at the terminal button, the synaptic vesicles release their neurotransmitters into the synaptic cleft. The neurotransmitters travel across the synapse and bind to the receptors on the dendrites of the adjacent neuron. Then the process repeats itself in the new neuron. That's actually a really good short summary of exactly what happens throughout the neuron. So for neuronal communication, I think it's really good to study that bullet point that I just read. Last one on the left side, reuptake is when the neurotransmitter is pumped back into the neuron that released it in order to clear the synapse. Moving over to the right, we start talking about drugs that can affect neurotransmitters. Agonist drugs mimic or strengthen the effects of a neurotransmitter. Antagonists block or impede the activity of the neurotransmitter. So you take an agonist when you need more of it and an antagonist when you need less of it. And remember, there's also reuptake inhibitors, which give the neurotransmitter more time to do its job. The central nervous system is the brain and spinal cord. The peripheral nervous system connects the brain and spinal cord to the muscles, organs, and senses in the periphery of the body. Within the peripheral nervous system, we have the somatic nervous system, which relays sensory and motor information to and from the CNS, the central nervous system. Then the autonomic nervous system controls our internal organs and glands. That's the non-voluntary stuff, the involuntary stuff. And within the autonomic nervous system, we had the sympathetic, which gets us ready for stress-related activities, and then parasympathetic, which returns us back to our resting state. Moving on to the brain, we have the cerebral cortex, which is the surface of the brain. Remember, it's covered in those bumps and grooves, and including the one big groove down the middle, which divides our brain into the two hemispheres. And the corpus callosum is the thick band of neural fibers which connects the two hemispheres. The limbic system is what processes emotion and memory, as well as smell. It's comprised of the hippocampus, the amygdala, and the hypothalamus. The hippocampus is responsible for learning and memory. The amygdala is responsible for tying emotional meaning to our memories and experiencing emotion. And the hypothalamus regulates our homeostatic processes like our body temperature and blood pressure. The cerebral cortex can be divided into 4 lobes. The frontal lobe, which is involved in reasoning, motor control, emotion, and language, and contains the motor cortex for our motor control. The parietal lobe, which is involved in processing the sensory and perceptual information, and one of its important parts is the primary somatosensory cortex. The occipital lobe is associated with visual processing, contains the primary visual cortex, that one's in the back of your brain. And then the temporal lobe, over your temples, is associated with hearing, memory, emotion, and some aspects of language. And the temporal lobe contains your primary auditory cortex. Makes sense that the part right above your ears is for hearing. That can help us remember that. The reticular formation, which is in the midbrain, regulates our sleep and wake cycles, as well as arousal, alertness, and motor activity. Also in the midbrain are the substantia nigra and the ventral tegmental area, or VTA. These produce dopamine and are critical for movement. And remember, the substantia nigra, when it gets depleted, is what happens during Parkinson's or causes Parkinson's. And the endocrine system is a series of glands that produce chemical substances known as hormones. We looked at the examples of hormones. In the summary, I did not go over the different types of neuroimaging. Basically, I ran out of space, but please do review the different types of neuroimaging, and mostly remember that the EEGs just give us the general electric activity, but they don't tell us where the activity is happening in your brain, and the fMRIs are our best bet for really knowing what's going on in the brain in a safe way.